Okay, in this lecture we're going to continue with the chemistry of life, part 5. And in this lecture we're going to learn about 4 molecules that are found specifically or predominantly in living organisms. All the molecules that we've discussed so far in the chemistry of life can be found in living organisms, and they can be found in non-living entities. But there are 4 molecules that are found predominantly in living things. We're going to discuss those in this lecture. Okay, there are 4 molecules that are found predominantly in living organisms. Those molecules the first one is going to be a polymer of these molecules called amino acids. So if we have here an amino acid, an amino acid, an amino acid. So if we have a string of these amino acids, a long string of these amino acids, we call this a protein. And that's the first molecule that's found predominantly in living organisms, a protein, which is a polymer of amino acids. The second molecule is going to be a polymer of nucleotides. And here we have, let's see here. It's going to be kind of tough to draw. We'll just put here nucleotide, nucleotide, nucleotide. Just a long string of nucleotides. And a lot of times that string is doubled, and it makes a double helix. And so we call this structure a nucleic acid. And nucleic acids, there are 2 nucleic acids in the human body, DNA and RNA. And so that's the second large molecule found in living organisms. The third molecule is going to be a long string of sugars. And sugars are usually, a lot of the sugars in the body, or the main sugar in the body, is a 6-sided structure. So I'm going to draw it like this, and then these sugars are linked to one another. And this is a long string of sugars. And so that long string of sugars is called a carbohydrate. Okay. And the very last molecule that's found in the human body, this molecule, there are lots of different types of this one, and it's not usually a polymer. One of them is made up of a 3-carbon sugar, or a 3-carbon structure, linked to these long fatty acids. And this is a fat. And all of these are carbons, so it's not really a polymer. Some of them have a structure that looks something like this. I'm not going to draw the whole structure, but it's 3 6-carbon rings and a 5-carbon ring. That's all of it I'll draw. Some of them are called, these are called steroids. And like this is cholesterol here. So steroids are also part of this last group. And steroids are based on cholesterol. And so we call these lipids. Okay. And there are others, but we won't talk about the others yet. Okay. And so, in the human body, there are several of these very large molecules. We have proteins, which are long strings of amino acids. We have nucleic acids, which are long strings of nucleotides. We have carbohydrates, which are long strings of sugars. And we have lipids, which can be either fats and oils or steroids. Now these 4 molecules, because they are very large, we call them macromolecules. We call them macromolecules. Macro means big, so these are macromolecules. Now the study of these macromolecules in the human body, we call this branch of science biochemistry. Biochemistry is the study of the chemicals in living organisms. A lot of times they include the proteins, nucleic acids, carbohydrates, and lipids, but they also include they also include the amino acids, some of the specific amino acids, some of the specific nucleotides, some of the specific sugars in the body. And so these are our macromolecules, and the study of these macromolecules, that's going to be biochemistry. And in the following slides, we'll look at these in detail. We'll learn about what are amino acids. We'll learn about the nucleotides. A few nucleotides we'll learn. We'll learn about the carbohydrates, and we'll learn about what's this 3-carbon structure here, and what are these fatty acids. And that's probably as far as we'll go with the macromolecules. Just a brief overview. And that's it for our macromolecules and biochemistry. Let us start with our first macromolecule and talk about proteins and amino acids. Before we do, there is a mnemonic that I use to remember the 4 macromolecules. If you look at the first letter of each macromolecule, P, N, C, L. PNCL, pencil. So if you remember the word pencil, you can always remember the 4 macromolecules. Okay, and that's it for the overview. Now let's talk about proteins. Okay. The protein is going to be the first macromolecule we investigate. Let's look at some examples of amino acids. There are 20 amino acids in the human body. 20 common amino acids, I should say. But what we're going to do now, I'm going to show you the basic structure of an amino acid. Then I'm going to show you 2 examples of amino acids. That would be good to know. Okay. Amino acids. They have an amino group, N, H, there's our amino group. They have a central carbon. They all have a hydrogen attached to that central carbon, and they have their second group, known as a carboxylic acid group, or your carboxyl group. And here on the amino acid, I'm going to make this one. This group here, we'll call it the R group. That's the part that makes amino acids different. That's the part that can change. So amino acids have several functional groups on them, and before we look at specific amino acids, we're going to define the functional groups here. Let's see here. This first one here, this functional group is called an amino group, and the one here, this is called the carboxyl group, or the carboxylic acid group. Let's see here. This carbon here, we call this the alpha carbon, and let's see, let's get another good color. This side, this R here, or this side chain, is called the R group, or the side chain. So let's label these before we continue with the structure of amino acids. Let's label these functional groups. Okay. Okay, I'm going to move this up a little. Okay. NH2 is known as the amino group. AMINO, the amino group. This carbon here is called the alpha carbon. We can use the symbol, the Greek letter alpha, or we can write it out alpha. The alpha carbon. This group here, well, we'll do this. Well, we'll go ahead and label it here. This is the R group, or the side chain of the amino acid. This is the only part of the 20 amino acids that changes. This is the side chain. And the last part of the amino acid here is the carboxyl group, or the carboxylic acid group. I'm going to call it the carboxylic acid group, and that'll become obvious in just a second. Many textbooks call this the carboxyl group, the carboxylic acid group. Okay. And so these are the different parts of an amino acid. Now, why is it called an amino acid? It's basically a conjunction of two words here. It has an amino group here. So it has an amino group on one side. It has the carboxylic acid group on the other side. And for that reason, we call them amino acids. And so this first slide here is showing us what an amino acid looks like. And on the next slide, we'll look at an example of 2 of the 20 amino acids. The amino acid. Okay, now that we know what an amino acid is, we never labeled this. Let's go and give it a label so we'll know this is a generic amino acid. And amino acids make up proteins. So here we have a generic amino acid. and we know why it's so named. Now what we'll do, we'll show you the structure of 2 of the 20 amino acids. Okay, so let's kind of get rid of a few things here. Okay, we don't need that. Okay, and we don't need this. So we have our amino acid here. Actually, let's put everything back here because I want to move this one. Okay, because at some point we may want to look at everything together. Okay, I want to move this over. No, that's not going to work. Okay, this is taking longer than I wanted. Let's get rid of that. Okay, now let's see if we can move this over. It's taking up more space than I wanted to, so I don't know how I'm going to do this. Oh, forgive me for just a moment. I need to do 2 things. I kind of need to make this smaller. Okay, that's good. So I know this is going to work. Now I will move this over here, and we're just going to look at 2 common amino. Maybe I put it down here. Yes, let's look at 2 common amino acids. But we don't need all of this. We don't need this. That's the only thing we need. And we're going to get rid of that for now. Okay. Let's go in here and look at some amino acids. Now on the first amino acid that we will show you, this R group changes. So I'm going to get rid of this R group, and I'm going to put the structure that actually goes there. Let's make it purple here. And so if we simply put an H here, we make an amino acid, and that amino acid is glycine. I hope I'm remembering my amino acids okay. And so this amino acid is glycine. Everything is the same except the side chain changed. So here we have glycine. And now we're going to make one more amino acid. This seems to be taking a lot of time, this part. So let's move this over here and make a whole nother amino acid. Okay, now let's change this one. Okay. And so here we're not going to have an H here, so let's get rid of the H. And let's give ourselves another functional group. Let's do this. This functional group, I'm just going to show you that some of them can be complex and some of them don't have to be. So let's give ourselves a let's still make it, what color do I want here? Still make it let's make it a bluish color. And so let's have CH2 SH. That's going to be our side chain. So this replaced the R. And so this amino acid is cysteine. Okay, and so here are two common amino acids. So that's our general amino acid up here, and two specific amino acids here. Glycine has an H replacing in the side chain, and cysteine has a CH2SH in the side chain. And remember, there are 20 amino acids, but we only need to know 2 of them. Let's put here the number of amino acids. There are 20 amino acids, 20 common amino acids. And that's it for the structure of the amino acids that I'll need you to know. We have glycine, the amino acid glycine, and we have the amino acid cysteine. Let's do something with these amino acids. Let's put them together. Let's start making a protein. So the first thing we want to do, okay, here are our individual amino acids. Let's also create a copy of these. Okay. Now what I'm going to do here, just so that we will see what's happening, I'm going to join this carboxylic acid group here with this amino group here. So I'm going to kind of, well, let's not do that yet. Let's pull these two closer together. Let's put these two closer together. Let's move this one over here. And let's move the cysteine over here. I'm going to do something right in here just so that we can see what's happening. The OH group, let's kind of get a really good color, because I need you to see what happens. I'm just going to change the color. Let's just do that. Let's change the color of this OH group. Let us make it, well, that's not going to really help me. Let's not change the color. Let's just highlight it. Let's highlight this OH group. Okay. Let's kind of highlight this OH group here. Yes. Here we are. Let's highlight that OH group here, and let's highlight let's highlight this H here. Let's see what would be a good color for highlighting it. Perfect. Perfect. Now, what's going to happen here? Okay, let's put these together and these together. Okay, now what's going to happen? You can see here we have OH and H, and the two of those together make water. They make water. And so what's going to happen here when we actually make this protein, we actually put these two together, we're going to actually cut off the OH is going to leave the first amino acid. Okay, it's going to leave that first amino acid. It's going to go away, and here the H is going to leave this amino acid. Okay, the H is going to leave this amino acid, and it's going to move away. And these two, well, let's just make the molecule here. And these two will end up making water, HOH. So that should be easy to see. But now a new bond is going to form between this C of the former carboxylic acid group and the N of the former amino group. And that bond is going to form between the two of them. And this bond, I'm going to make it kind of conspicuous. A bond is going to form between them. And now we have a new, we have a bond between two amino acids. Now that bond is given a special name, the molecule that leaves is given a special name, and the new structure that's formed is given a special name. But first, let's move the HOH out of the way. Let's move the water. I have to find the water first. Okay, and there it is. Got to find the water molecule. Excellent. Okay, here's water. So I'm going to move water out of the way. So we formed water, and we formed this structure here. Okay, let's start naming some things. This bond, the bond that forms between, well, actually, let's just stop here on this slide. And so we see what we formed. Let's just get a review. We have glycine, we have the amino acid glycine, and we have the amino acid cysteine. The two of these amino acids came together. We lost OH from the carboxyl group, we lost H from the amino group to form water. Then a bond forms between these two amino acids. When we come back, we'll learn what this whole structure is called, what that bond is called, and what this HOH is called. But we already know. Okay. And that's it for this slide. Let us now label this structure here. Okay, the first thing that we're going to label is this bond, the bond that forms between glycine and cysteine, or the bond that forms between two amino acids. Now, whenever two amino acids join in a protein to make a polymer, it's always the carboxyl group joining with the amino group. This bond here is called a peptide bond. Peptide, strange word. P-E-P-T-I-D-E. This is a peptide bond. Okay. And the structure that's formed when amino acids join together, that structure, if it's 2 amino acids, 3 amino acids, 4 amino acids, it doesn't matter. That structure is called a peptide. So what we have here is a peptide, a structure made of more than one amino acid, or amino acids joined together by a peptide bond is called a peptide. And since this peptide is made of 2 amino acids, we can give it another name. We can call this peptide a dipeptide. So a dipeptide equals 2 amino acids. And that's what we have here. So let's get some general terms. Let us identify a few general terms. Now, if we had a peptide, if we had a structure that was made of three amino acids, that would be a tripeptide. And that makes sense. Okay. Well, let's not even do 4 amino acids. Well, 4 would be a tetrapeptide or a quadrupeptide. If it's a structure that's made up of anywhere between 10 to 15 amino acids, 10 to 15 amino acids, because these structures are sometimes common, we call this an oligopeptide. Oligo means a few. Oligopeptide. And if we have a structure that's made up of many amino acids, like a protein, if we have many amino acids, we call this a polypeptide. And we'll talk about this more in later slides. And so here we have, review. Glycine is an amino acid with an H side chain. Cysteine is an amino acid with a CH2SH side chain. When these two amino acids come together, the C of the carboxylic acid group joins with the N of the amino group, and we lose this compound here, HOH. And HOH, of course, is water. And that is what we lose. The bond that's formed between these two is called a peptide bond, and this structure is a peptide. If it's just 2 amino acids, the peptide is called a dipeptide. If there are 3 amino acids, the peptide is a tripeptide. If there are a few amino acids from 10 to 15, we call it an oligopeptide. If there are many peptides, we call it if there are many amino acids, we call it a polypeptide. So the main point here is the peptide bond. And that's it for the peptide bond. Let us draw a protein or a string of amino acids. So let's just make ourselves a nice string of amino acids. Okay, let's go here, draw ourselves a string of amino acids. We'll make the amino acids different colors. Okay, just going to make ourselves a nice string of amino acids. Okay, here's our amino acid. These are our amino acids. Okay, let's join them with a peptide bond. Okay. Here's our peptide bond holding the amino acids together. Now, when you're just concerned with the sequence of the amino acids, this structure is given a name. We just call this the primary structure of a protein. And so this is the protein's primary structure. This is the primary structure of the protein because it's just the amino acids. Okay. Now, I don't want to have to draw all of this every time. So in the future, rather than drawing all of this, I'll just draw this for the string of amino acids. Okay, and so that's the primary structure. Let's look at another type of protein structure. Let's look at the second type of protein structure. Sometimes that primary structure can start to fold on itself. Not completely fold, but you can start to get these helices in the protein structure. Okay, or the protein structure can do this. It can form two strands can get together to form these folds, or like pleats. Some of it can remain not folded at all. And so whenever you have these two structures here, or you have the either the pleats, these folds here, you can have this structure here, or these foldings, or these pleats. These are given names, and let's get those names first. This fold is called an alpha helix. And this structure, the second structure, is called a beta pleated sheet, because it's like a sheet that's folded, a beta pleated sheet. Of course, it's made of two. The strand has folded on itself. And so these two structures are called secondary structures of the protein. This is secondary protein structure. Okay, so we have secondary structures when some of the protein is not folded and some of the protein is in alpha helix or beta pleated sheet, and the rest of it is not folded. So this is the second level of protein structure. The third level of protein structure is one in which the whole protein is folded. And so you have the protein folded in on itself, and in that folding you can have some alpha helices, you can have beta pleated sheets here. Protein can be more folded. You can have more alpha helices, more folding of the protein, more beta pleated sheets here. And so the whole protein is folded, but it's just one protein. And so we call this type of structure tertiary structure. This is the tertiary structure of the protein. Okay. Now, for the next one, I need to kind of condense this tertiary structure. And so this is a completely folded protein. Now what we're going to do for this last, well, let's not do the last structure yet. Now for this tertiary structure, that's okay, we'll leave it like it is. But now on this last one, what I'm going to do, I'm going to take the tertiary structure of the protein. I'm going to make myself there we are. Okay. I'm going to take this tertiary structure. Okay. And we're going to move it over here. I need to make it a little smaller so that I can do something with it. Okay. Here's our tertiary structure of the protein, and I kind of need to another one of those. I want to give it another color here. Let's make ourselves a blue one. Let's make ourselves a red one, and let's make ourselves a green one. Okay, that's good. Now for the last level of protein structure, what happens is several of these tertiary structures come together to make a more complex protein. And so this type of structure is known as quaternary structure. And we'll continue this on the next slide. Let us continue where we left off here. We have different levels of protein structure. Okay, the first one, when you just have the sequence of the amino acids, that's primary structure. We don't have structure spelled right, so let's fix that before we continue. Okay. Okay. That's primary structure. Now, with primary, if you just have primary structure, we call this, if you just have primary structure, we call this it's just a polypeptide. It is not a protein yet. It is simply a polypeptide. It is not elevated to the level of a protein. So when you just have primary structure, you just have a polypeptide, or many amino acids joined together with a peptide bond, with peptide bonds. When you just have secondary structure, you still just a polypeptide. Only when the protein is functional and it's folded correctly do we call it a protein. And so here, this structure is a full-fledged protein, because it's completely folded. And of course, some proteins can join together to give this fourth level of structure, and this is called quaternary structure or quaternary structure. Quaternary structure. Okay, and sometimes that's written as a 4' or 4 superscript O. And tertiary structure is sometimes written as 3. Primary 1, secondary 2. And so these are the 4 levels of protein structure. On the next slide, we will talk about, we can see here the protein has to fold completely, or the polypeptide has to fold completely to become a protein. And so what goes on here is a folding. So a folding has to take place. And what we'll do on the next slide, we'll find out what that folding is called. And sometimes we can actually unfold the protein and cause it to go back this way. And we'll find out what that unfolding is called. But we'll do that on the next slide. So here we have the 4 levels of protein structure. Let us draw a folded protein here. Okay. Here we have, or not yet, but let's draw a folded protein. And so This is our folded protein. Let's give it some secondary structure, some alpha helices, more foldings. Let's give it some beta pleated sheets here. Okay, here is our folded protein. Okay, let's move it to the side just a little. And let's make it a little smaller. It's kind of big. And so here we have our folded protein. Now, this normally happens outside of the body, but sometimes what you can do, if you want to unfold that protein, you can introduce some components to this system. Okay. If you add, let's say either you could add a lot of heat, or you could change the pH. That will cause that protein to unfold. And when the protein unfolds, of course, you'll simply have a long chain or polypeptide. And so here, this is a functional protein. So this is a protein. Now the protein can also be called a folded polypeptide. Okay. And over here we have an unfolded protein. Here we have an unfolded protein. And the unfolded protein can simply be called a polypeptide, and it only has primary structure or secondary structure. So this is just simply a polypeptide. Now let's talk about the process. The unfolding of a protein, or moving this direction, the unfolding of protein is called denaturation. So when you unfold a protein, that's called the denaturation of the protein. D-E-N-A-T-U-R-A-T-I-O-N. That's the denaturation of the protein, or the unfolding of the protein. And of course, when you go back in the other direction, when you actually fold the protein, that's called naturation, or folding of the protein. It's just called naturation. This word is not really used a lot. I never hear anyone say this protein naturated, but it's the opposite of denaturation. But this term is not used. Normally we just say the folding of the protein. Okay, and that is all I have to say about the denaturation of a protein.